My name is Redmond Smith, and I will be moderating the two talks today. So we had over 120 people register, I hear, and many of you are coming online already, I can see. So today's seminar is hosted by the HIRI in Wuerzburg, the HIRI being the Helmholtz Center for RNA-based Infection Research, which is the first institute in the world to focus solely on the role of RNA in infection. So Wuerzburg in Bavaria, in Germany, is a great home for this relatively young institute, as it's a major hub of infectious disease research in Germany. So we are currently 8 groups investigating various aspects of bacterial and viral pathogens, and we're at a very exciting phase of the institute. We're still rapidly growing, continually looking for talented researchers who are interested in RNA and infection on all levels, from PhD to group leaders. So if that sounds like your cup of tea, you should check out our website for jobs, or even, and I'm not sure I dare say it, one day pay us a visit. So today you'll hear from 2 of my colleagues, excellent scientists, Junior Professor Neva Caliskan and Junior Professor Mathias Munschauer, who are going to talk about their work on RNA viruses. So the presentations are scheduled to be 25 minutes each, with 5 minutes of questions. I would recommend you type your questions into the chat and I'll deliver them to the speakers, but I will also be but if you have a burning question, I would recommend you type it in. So the talks will be put on YouTube, most likely tomorrow. So if you know somebody who wasn't able to hear us live, they can still watch the presentations later. And I would appreciate if you pass on this information to your colleagues. So before starting, I'd like to thank Tim Schneider, our scientific coordinator, for helping to organize these seminars today. And with that, I'll introduce our first speaker, Neva Caliskan. She studied molecular biology and genetics at the Middle East Technical University in Ankara, Turkey, and worked as a visiting scholar in 2005 at the EMBL in Heidelberg. She received her master's degree at the International Max Planck Research School for Molecular Biology in Göttingen in 2009. And after completing her PhD in Marina Rodriguez's group at the Department of Physical Biochemistry at the MPI, she continued working as a postdoctoral fellow first, and then as a project leader at the same institute. Now, since 2018, she holds a junior professorship at the medical faculty of the University of Wuerzburg, and now heads her own research group called Recoding Mechanisms in Infections at the HIRI. And Neva's group is spending a lot of scientific energy into understanding how viral and host proteins modulate recoding in viruses. And this is what you'll hear about today. So with that, let's get started, Neva.
Thank you so much, Redmond. I think you covered also my first 2 slides. I can save that time. So hello, everyone. Good afternoon, good morning, or good evening. I can see a number of the participants is increasing, but let's just get started. So as Redmond just mentioned, I'm going to tell you about RNA binding viral protein that acts as a viral translation gene switch. And I'm very happy to be invited also as a speaker. So we'll just briefly give you an overview of what we do in our group. And if time allows, I also would like to mention a few more words on our recent ongoing work on the coronaviruses. So viruses are active research topics in our institute, and especially with the outbreak of the pandemic, we started to put more efforts in understanding viral mechanisms, RNA-mediated mechanisms. Mainly viral translation events are very much hot topics in my team, and we want to understand interplay between viral RNA elements and proteins, and trying to understand how various RNA elements interrogate RNA function. Viruses like the cardiovirus, which is the topic of today's talk, and the coronaviruses share similarities. They both carry their genetic information in the RNA. And once a virus enters a cell, it requires the host ribosomes to translate its RNA. This is important for its replication. The coding regions of these RNAs also contain specific RNA elements, as you could see here, which leads to ribosome pausing and can stimulate translation of various peptides from the same RNA molecule. This can happen through read-through, frame shifting, or hopping. Although we learned a lot about these cis-acting regulatory elements and how they work, we still know very little about the function of these transregulatory elements, whether there are transregulatory elements and the function of these elements in these processes. So we all appreciate and started to appreciate even more that RNA exists rarely alone in a cell, and that's also true for recoding elements. Specific host proteins, so viral proteins, even small RNAs can be involved in regulating the level and the timing of recoding events, and molecular details of this regulation is not very well understood, as I just said. We are interested in a frame shift mechanism. This is a universal recoding strategy to access multiple operating frames. These sites contain usually structured RNA motifs just at the site where this event happens, which impede ribosome movement, and that generates a time window for codon-anticodon interactions to be broken and repairing to happen. What is interesting is, for instance, you would see here the cognate interactions are maintained, but after this, in the overlapping codon, the meaning of the code would be completely altered. So protein with a different function or a different sequence and a different function can be produced. And this, in a way, opens up a hidden code in the genome to produce various peptides from the same RNA. And unlike random frameshift events, these could also be functional. I'm giving more, now I would like to give you more information about the cardioviruses. For those of you who are unfamiliar with viruses, there's a single long open-reading frame that would be basically producing a large polypeptide that would be proteolytically cleaved by the viral protease. And in addition to the internal ribosome entry site or stop-go translation, recently the group of the Ian Brierley from University of Cambridge, also our collaborators, discovered this frameshift event that was modulated by the 2A protein that's produced here, and translating ribosomes would basically stop here after the translating the 2B star frame in the alternative reading frame, -1 reading frame. And this way, and this way translation would proceed translation wouldn't proceed further to translate the polymerase and the protease, and this would downregulate the level of the viral proteins. This protein was actually known for more than 20 years, but it was also shown to be associated with eukaryotic ribosomes, and it's involved in shutdown of cap-dependent translation. What's new is that on elongation, its function was less known, and what Navdin et al. Has shown on 2017 was the protein actually induces significant levels of frame shifting by up to 70%. But how does the protein interacts with various frameshift stimulatory elements, and whether it also interacts with translating ribosomes was our interest. Before I go into the details of the in vitro characterization, I would like to show you the crystal structure of the 2A, kindly provided by our collaborator, Dr. Chris Hill, from the group of Ian Brierley. The 2A protein has shown no structural homology to any other RNA binding protein in the PDB. That was already quite interesting. So that's a novel RNA binding fold. But we could immediately recognize the high degree of flexibility of the beta strands and the surface loops, particularly those containing arginine and lysine residues. And these arginine residues were important for inducing frame shifting. It was also previously shown that 2A can non-specifically interact to various RNAs. So it could be due to this great flexibility, but the other question is whether ribosomes interact non-specifically with the 2A, or RNA interactions are specific or not. Despite its structural flexibility, actually, the EMCV or the TMEV RNAs can specifically interact with the cardiovirus RNAs. To understand the interplay between the 2A protein and its various interaction partners, we employed the highly sensitive microscale thermophoresis technique that works with capillaries, and it's very efficient in sample consumption, so I can only recommend it. And we were one of the few groups that employed this to understand RNA protein interactions. We have seen with the wild type RNA that the RNA interacts with nanomolar affinities, and you can see here comparison of EMSO results and the truncations of the 5' end, for instance, that completely inhibits its interaction with the 2A protein. When we truncated the 3' end of the RNA, we again see affinity, high affinity binding. And if both of the 5' and the 3' end was truncated, interaction was again impeded. So this shows these are important regions, especially the 5'. And when the loop region was mutated, which was also shown previously to inhibit frame shifting, when these loop mutants were the crucial elements for frame shifting, then again, interaction of 2A was significantly reduced. Last but not least, the slippery site mutants were generated to show whether slippery site actually is involved in this interaction, and actually it had no role in this particular RNA structure. To show that the effect we observe is specific for the ribosomes as well, we isolated eukaryotic 40 and 60S subunits, and we were able to show that the 40S ribosomes do associate with the 2A at very high affinities actually, and we didn't observe a high affinity interaction using the large subunits purified. So this was actually independently confirming the supported role for 40S interaction by Groppo et al. So we then asked whether we can demonstrate the same effect in bacterial ribosomes, because both my former work and others have shown viral frame shifting actually can be studied using bacterial ribosomes. For that, we just use our 3050S purified ribosomes, and we're able to demonstrate that basically 30S show a very high affinity, whereas it's moving too fast. Whereas 50S again show barely any specific interaction. So with that, we decided to check bacterial and eukaryotic frameshift levels. We were able to, using a frameshifting reporter with mCherry and GFP, basically frameshifting would produce the mCherry GFP fusion protein, whereas the GFP signal would only be produced in 0 frame. And using the system, we were able to recapitulate what was shown by our collaborators formally, that in vivo eukaryotic frameshifting levels was reaching about 60% in the presence of the 2A. On the other hand, 2A was still able to produce significant levels of frameshifting by more than 20%, and no cellular factors were necessary for its function. This means bacteria was actually a good model system. Due to its simplicity, we just decided to look at this more in detail. So what we did was building initiation complexes, programming them by mRNAs encoding for the frameshifting site, and using these, we measure again binding affinities, and we have seen initiation complexes do have significant affinity to A. And what was more interesting is, surprisingly, ribosomes can also, vacant ribosomes can also interact with the 2A. We now cannot conclude whether this interaction is actually with the ribosomes is protein mediated or RNA mediated. But our collaborators had a great tool. Basically, Chris then carried out some cryo-EM analysis in Cambridge to look at these interactions in more detail, and that revealed very interesting hits on its function. Basically, we had this ribosome, initiated ribosomes at 2.7 angstrom, and it was interesting to see not a single copy, first of all, but 3 copies of 2A can associate with the 16S rRNA of the 30S subunit. Then Chris compared this to published 70S pre-translocation complexes or to 70S EFTU, bound with EFTU, and we were intrigued to see in 2 copies of the protein would clash with the factor binding site for EFG as well as the EFTU. This means this can actually explain how the protein can prevent translation elongation process. And similarly, probably also the initiation factor binding, which is all GTPases bind to similar positions, basically. Coming back to the RNA, this still doesn't tell us why frame shifting is stimulated by these interactions with the ribosome. So we decided to dig more into this, and we studied the mutations that were shown to inhibit frame shifting, namely the CUC and this UUU mutations. I would like to point your attention to those mutations in the loop that inhibit, basically, although they inhibit binding, barely any difference was seen between their predicted structures with the wild type and the CUC mutant. In reality, we do observe a strong difference in their binding affinities, though. This region is definitely then involved in the interaction with the 5' proximal region of the hairpin. Therefore, we decided to look more into its dynamics. When we look at the predicted pseudoknot and stem loops of the structure, there was some heterogeneity, and it was difficult to understand what does it mean, and to dissect this heterogeneity of RNA molecules, we employed the single molecule techniques, the optical tweezers, and this was done by a very talented PhD student from my group, Lukáš Pekarek. Now I'm going to tell you a little bit more about his findings. So we basically hybridize the cardiovirus frame shifting RNA to single-stranded DNA handles and tether them with streptavidin or digoxygenin conjugated beads, as illustrated in here. The beads were then trapped with the laser beam, and this allowed us to mechanically manipulate the RNA region of interest. So in this case, by precisely stretching at a constant rate, we were able to monitor unfolding, and when we release the force, we can nicely see the refolding. And I'll show you just 2 example traces to that. These are example force distance curves. So you increase the force and then measure the distance at the same time. In the first population on the left side, we see that RNA unfolds in 2 steps. So basically a rupture would be seen by the force wouldn't change while the distance would increase. So this means that's an unfolding step. And this was in line. 2-step unfolding and refolding can be seen in the blue trace. So this shows that unfolding happens in 2 steps and refolding happens readily in this case. This is in line with the stem loop, for instance. On the other hand, some molecules show a higher unfolding force and barely recognizable low force unfolding events, but major unfolding happened at much higher forces. And interestingly, they show a large hysteresis, meaning they were refolding not readily, but at much lower forces. And that's a behavior common to many highly structured RNA molecules like G quadruplexes or some pseudoknots, which was shown previously by other groups. So once we know our RNA exists in various conformations, we wanted to understand on the population level how our RNAs do behave. So we see several intermediates. One was a very low unfolding molecule, a very low force unfolding molecule. This shows these 2, and another one showing 2-step unfolding. These were in line with relatively weak RNA stem loops with short stem loop region, basically. Another one, this was a major state. This showed unfolding occurring at less than 10 piconewton, and the second step occurring at 20 piconewtons. This was basically in line with the stem loop with the bulge here. And a state 4, which we call was a stable pseudoknot-like conformation that was previously, I've shown you the refolding trace didn't happen readily. So this was a stable conformer population that shows no unfolding steps under the measure. And wait a second. This showed a very high unfolding step. And lastly, there was another state that showed no unfolding at the highest unfolding forces that we applied. Overall, the unfolding and folding behavior was interesting. So we then decided to look at that on a global level. Basically, we observe in the presence of the 2A protein, increase in the higher force, basically upper more than 20 piconewtons, that was increasing, and the very high force population was also increasing. This is state 4 and state 5, but in the presence of the 2A, that was the major state that showed the difference. Strikingly, populations of molecules in the low force, basically state 1 and 2, decreased in the presence of the transacting factor. Looking at the CUC gave us clues what these conformers can really be. Basically, we see no real difference in the population profile compared to the wild type, compared to the in the presence and absence of the 2A. Without the protein, there were only more minor changes in the normalized distribution of the unfolding forces. Basically, slightly they moved to higher force intermediates, but there was no significant difference compared to the wild type. I mean, compared to the wild type where we observe really high force intermediates. State 4, the pseudonode-like state was completely absent in the presence of the 2A, as well as the state 5, which was the major dominant population in the presence of the 2A. This was also completely missing. This was also consistent with the predicted, and the majority of this would exist in state 1 and 3 basically, and that was consistent with our observation that the predicted stem loops would be able to form in the CUC mutant. However, it doesn't transform into the highly stable states. To put these together, 2A may preferably bind to the semi-folded intermediates, which is the state 1 and 2 in our case, and 2A might help them to remodel them into a highly stable conformer that is state 5, that differs from the other states that we have observed. And in the absence of the 2A, that state is not dominant, but in the presence, we see more and more that's the most dominant state. Taken together, this result suggests that 2A binding stabilizes this secondary structure and it increases its interactions and mechanical resistance to unfolding. Well, that is now our current working model. We think that EMCV transitions between various conformers, EMCV RNA transitions between various conformers and this is important for its function. During the infection, the concentration of the 2A would increase, and this would allow it to selectively bind to the stable pseudonode and stabilize this pseudonode structure, which is like an on-off switch. Basically, the pseudonode-like stable conformation is like acting like a frame shifting switch, and interactions with the 5' proximal site and the loop of the frame shift element is important for this function. Stimulation of frame shifting would lead to production of less of the enzymatic viral proteins later during infection, so this can have important implications during lytic infection. On the other hand, the protein 2A can also bind to the small subunit, and binding at the region where translational GTPases would be associated to the ribosomes and elongation steps. So 2A is then a multifunctional translation regulator according to our analysis. Now, in the last minute, I would like to just give you an idea whether we had, by the outbreak of the coronavirus, we have been trying to understand whether this actually can be similarly regulated, whether coronavirus can be similarly regulated. And for that, we have employed, first we came to realize that previously published RNA protein, which is shown to be regulating HIV frame shifting, it was an interaction partner identified in my in the direct interaction screen in our colleague's screen, Mathias Munschauer, which will just take over the stage. And he came to me and then mentioned that this protein was one of their hits, and we just decided to test it in our dual fluorescence system, basically using the very same system for the SARS coronavirus. We were able to show that RNA or the shiftless protein does influence frame shifting, and here is the results of this frame shift analysis. Basically, in the control, in the presence and absence of the transacting factor, we see no difference. However, on the HIV frame shifting side, this frame shifts about 5%. We were able to see the same 30% decrease, which was previously reported last year by others and Wang et al. And then we were able to also show SARS coronavirus follows a very similar route. These results are just published, so you could check them out. On something we have also realized is the shiftless also behaves similarly on sucrose density gradients. Basically, it's associated with the 40S as well as the 60S and the 80S. This means host and the virus-mediated regulation of recoding events can present a common strategy in viruses. So we are inspired then to see if other factors can be involved in this regulation. For that, we are moving forward and identifying further interesting RNA binding factors that can be involved in that by using a specific in vitro RNase antisense purification technique, using infected and non-infected lysates, and characterize them using proteomics. And probably I'm going to then tell you next in the next meeting what are some of the interesting results in the next meeting. So with that, I would like to finish quickly and would like to acknowledge my brilliant lab members whom made this work real, and that was really great work done by two of the talented PhD students, Anuj and Lukasz, the cardiovirus story, as well as Dr. Chris Hill, Southern Upton, and Andrew Firth from and Ayn Riley from University of Cambridge. They have been amazing collaboration partners, and also would like to thank Lumix people for their help and suggestions with the single molecule analysis, and would like to thank my past and present mentors and the funding resources. So please feel free to get in touch with me if you're interested in our work, and I'll tell you more about that in detail. With that, I would like to finish, and thank you for your interest.
Thanks, Neva. That was a really wonderful presentation, super clear. So what I'm going to do now is I'll look in the chat for questions. There aren't any at the moment, so I guess you're all furiously typing away. And I'll also try to scan through. If anybody wants to raise their hand, then they can ask the question directly, but it's a little bit tricky because there are so many of you. Oh, we have our first question from Jennifer Miller. Did you ever try the optical tweezer experiment with the CUC mutant? It could be that the pseudoknot is abolished with the mutation. Good question. Neva, you're on mute there.
Yeah, thanks for the question, Jennifer. We have done the analysis, the same analysis using the CUC, and we didn't actually see any of the stabilized states. So whether I should share the screen again, maybe. I mean, it wasn't present basic. Stop share. We didn't observe any stabilized intermediate in this case. So this means that the state 5 was really the stabilized intermediate, and its absence in the CUC mutant also showed that state 4 and state 5 wasn't changed. This shows that the conformationally stable states were actually these two, and these are absent in the CUC mutant.
We have another question from Kurt Gustin. Sorry if I missed this, but any idea what the role of 2B star is?
To my knowledge, I mean, perhaps Andrew or Chris can comment, but I don't know any known function other than downregulating the level of the downstream genes, which is the polymerases. So their enzymatic proteins are downregulated. But 2B is a relatively short peptide that is not known to have any specific function so far.
We have Ina Huppert's very nice presentation. Do you think the behavior of the UUU RNA mutant is unique, or would other mutations cause the same effect in terms of the pseudoknot?
That's a very good question, Ina. Hello, by the way. And the UUU mutant is specific because every single nucleotide was mutated, and this region was found by our collaborators to be really important for the frame shifting. And that can actually interact with these, on the protein side, these arginine residues were found to be important for its interaction with the RNA. So for that reason, we think that UUU specifically binds or kind of 2A mediates its interaction with the 5' proximal region, and that's how the interaction is leading to frame shifting that can, forming a more stabilized intermediate. But it's interesting. I mean, I don't know whether immediate proximal nucleotides to this UUU, whether they might have an effect, but I'm sure that was already studied.
Then we have another compliment from Charles. Great talk. In the EMSA, there seems to be two distinct complexes with 2A. Any idea what the differences are?
Well, that was done by our collaborators at the EMSA part, and they have shown this also previously, that RNA, this can actually be multiple 2A molecules. It can be, and this is actually also confirmed now by our ribosome analysis, that 2A basically dimerizes or forms trimers or even tetramers. It's possible. So this is at the increasing concentrations that was observed by their EMSA.
And I think I'll get a question in, Neva. So do you know what happens if you remove the 2A? I mean, is the 2A refolding the RNA into the pseudoknot? If you take it away, will you still have this increased pseudoknot population, or will it go back to these stem loop forms?
How can we take it away? I didn't get that, sorry.
Well, I guess you could just try digesting it away.
On the
Yeah, when you add the 2A and then remove it, do you still see an increase in pseudoknot formation? Or does the 2A still need to be there?
The protein is really prone to aggregation. I think it wouldn't be a problem to have this protein misbehaving. It has been a very difficult protein to work with at high concentrations. We never actually tried degrading it, and I'm a bit afraid in putting that in the single molecule system because it might be difficult to wash the proteins away. Yeah. So didn't try it, but it's an interesting idea.
Okay. Lars has got one last question. Could the binding to the rRNA compete with the binding to the mRNA during frame shifting?
Excellent question, Lars. Thank you. I mean, that's actually interesting. We still don't understand, and we are carrying out further kinetic analysis to really reconstitute the whole system in vitro to really understand what happens at the frame shift site, really, because we see for the binding affinities, we don't see, we see that the binding affinity to ribosome is actually slightly higher than that of the full-length mRNA, or to similar extent. It is possible that ribosome can compete, but what I kind of favor is that the ribosomes associate with this protein, and 2A can be hanging there. Either protein is brought into the ribosome by its interaction of the RNA molecule, or it is already bound, but not causing a very significant decrease in the rate of the translation elongation. Although it slows it, it's not completely preventing that. But as it reaches this pseudoknot or the RNA hairpin, then it is favoring the formation of this pseudoknot, and ribosome can then be bringing these auxiliary factors into the site of frame shifting. So that's kind of my favorite hypothesis at the moment.
And then I think there's a related question. In the interest of time, this will be the last one. This is from Junji. Is it correct to think that 2A has two functions, binding to ribosomes and binding the pseudoknot RNA? Have you found mutations that decouple these two binding events?
Great question. Again, we think that at the moment it works like it has different functions on normal translation and pathogenic conditions, and that the binding to the pseudoknot RNA is particularly important to regulate or regulate the levels of the polymerase. It has a clear biological function in that sense, but protein can also inhibit 4TS initiation step of translation. And now what we also shown by the EFTU and EFG binding of the protein to where the other GTPases can also interact with the ribosome. This also shows it can be actually inhibiting translation rates, overall translation rates. Whether we can find mutants that decouple these two binding events, if I remember that correctly, the region of interaction for the RNA was different from the region of interaction on the ribosome. The protein was showing different surfaces was interacting with the ribosome than with the RNA. So this also can show that it's probably interacting first with the ribosome and then brought into the pseudoknot or the RNA structure. So these mutants would be interesting to test to decouple these two functions.
Fantastic. Thank you, Neva.
Thank you.
That was great. I guess Mathias can now share his screen if he's still there. Yeah, we got you. So I'll just introduce Mathias now as our second speaker. So Junior Professor Mathias Munschauer, he started working with RNA and RNA binding proteins as an undergraduate student with Thomas Tuchel at the Rockefeller University in New York, completing his undergraduate thesis research on the RNA binding protein FMRP. He then joined an international PhD exchange program jointly operated by the MDC in Berlin, working with Markus Lanthaler and the New York University, working with Christine Vogel. In the Lanthaler lab, Mathias pioneered technologies to capture mRNA-bound proteome and measure protein occupancy on mRNA transcripts for the first time. For his postdoctoral research, he joined Eric Lander's group at the Broad Institute, where he identified novel long non-coding RNA-dependent topoisomerase complexes and dissected how NORAD contributes to genome stability. So he joined the HIRI relatively recently in July 2019 as head of his own independent Helmholtz Young Investigator group called Long Non-Coding RNA and Infection Biology. And today he'll tell you about some of his recently published work on the SARS coronavirus. So take it away, Mathias.
Thank you, Redman. This is really a fantastic opportunity to present the work of my group at this platform. We're generally interested in RNA viruses, and this of course includes the SARS-CoV-2 coronavirus. Our focus is the viral RNA by most part, and how the viral RNA interfaces with host cell RNA biology, really. A quick look to get started on the coronavirus replication cycle. Upon infection of a host cell, coronaviruses deploy a translation-ready RNA molecule that directly engages host cell ribosomes to produce the RNA-dependent RNA polymerase complex. This enzyme then uses the viral RNA genome as a template to produce negative sense RNA and produce subgenomic RNAs from that through a process known as discontinuous transcription. These subgenomic RNAs code for structural proteins of the virus. They're being translated at the ER, and they're also being incorporated into the ER membrane. They assemble with the nucleocapsid protein that binds to replicated copies of the viral RNA genome and forms novel virions that are then released and are able to infect new host cells. This is, of course, a very simplified view on the replicative cycle of SARS-CoV-2. However, I wanted to highlight the processes in pink here as candidate processes for regulation by host cell RNA binding proteins because they're centered on RNA processing. Human cells encodes almost 2,000 RNA binding proteins that have been identified over the years. And they regulate really every aspect of RNA metabolism, ranging from capping to polyadenylation, stability, transport, and localization. Since viral RNAs very much mimic host-encoded mRNAs, it's easy to imagine that these viral RNAs are also subject to regulation by these host RNA binding proteins, and this, by extension, would affect the viral RNA functions that we have seen on the previous slide. So my lab is basically asking a very simple question, which was at the time, what are the host cell proteins that directly bind and regulate these viral RNAs in infected human cells? And we have a particular approach to investigate what these proteins are. This is known as RNA antisense purification, and it's based on a very simple principle. And that is, if you know the sequence of your RNA of interest, you can very easily design antisense oligonucleotides that can hybridize to your RNA of interest. These antisense oligos can be biotinylated, allowing you to perform hybrid capture experiments on these target RNAs. There's many different flavors of this technique out there. In my lab, we use the technique developed by Mitch Goodman and Eric Lander, RAB-MS, and we like it very much for three reasons. And I'm highlighting these reasons here. The first is we're using, or the technique uses UV cross-linking to selectively only cross-link direct interaction events. With UV cross-linking, you do not get protein-protein or RNA-RNA interactions cross-linked. So we form inside cells that are undergoing infection, we're forming these covalent UV-induced bonds between protein and RNA. Second important principle is that we're employing denaturing purification conditions. We're effectively solubilizing the entire cell, and under these protein denaturing conditions then provide our antisense oligonucleotides, allow them to hybridize and capture our RNA of interest. Third important principle, we're comparing the protein content of these RNA purifications quantitatively using mass spec. And we're doing that relative to a known control complex. So this serves two purposes essentially. We can say something about the specificity of the interactions that we detect because we compare them to another RNA that is unrelated in its function. And it also basically serves as an internal control that tells us this experiment worked and we are able to recover known physiological interactions with this approach. So we did this for SARS-CoV-2 and we sequenced the protein cross-linked RNA fragments after these RNA antisense purifications. And what we saw is that in SARS-CoV-2 purification, 90% of the sequence reads mapped to the SARS-CoV-2 genome, which was satisfying purification of an endogenously expressed control RNA, RMRP, also from infected cells, close to 100% of the sequence read mapped to the human genome. If we look where do these protein cross-linked RNA fragments come from, they seem to be distributed across the entire length of the viral RNAs. We're sampling interactions not only from regions that are highly expressed, for instance, the 3' end, but across the entire RNA. Looking at our control first, this serves to demonstrate that this technique can really capture known physiological interactions, as I have pointed out earlier. Our control RNA here is the RMRP RNA, which is a endonuclease complex that is involved in ribosomal RNA processing. And we have atomic resolution knowledge about the protein RNA interactions of this complex, thanks to this beautiful cryo-EM structure shown on the right. Now, if we look at the proteins that we find to enrich in RMRP purifications, those are exactly the proteins that we see here to make direct RNA contacts. So relative to these, to this RMRP complex, we're now quantifying proteins that are enriched in SARS-CoV-2 purifications. And this is a scatter plot of two replicate experiments that we did. Viral proteins are shown in pink here, human proteins in purple. We detect in total 15 viral proteins, including 10 of the 16 non-structural proteins. Many of these proteins are known RNA binders. And for instance, the NSP12 protein, which is the RNA-dependent RNA polymerase, shows up quite strongly enriched. Similarly, the endoribonuclease NSP15, but also two components, NSP3 and NSP6, which are candidate components of a newly described pore complex in double membrane vesicles that form in infected cells. And these proteins appear to be important for the export of newly replicated or transcribed viral RNA. These show up quite strongly enriched as well. There are also surprises here, for instance, the spike protein, which we did not expect to see, but it seems to cross-link to viral RNA quite robustly. Of course, from our perspective, the most interesting proteins were the human proteins that directly bound to viral RNAs. We defined an expanded interactome. Those contains more than 100 human proteins that bind to viral RNA, as well as a core RNA interactome, which has about 55 proteins that are strongly significantly enriched in these purifications. I told you earlier that RAP-MS is specifically designed to identify direct interactors. We confirmed this subsequently using a complementary approach, cross-linking and immunoprecipitation. This is essentially using the same principle of using UV cross-linking to form covalent bonds between proteins and RNAs in infected cells. But now, instead of pulling on the RNA and finding the protein, we're pulling on the protein and sequencing the RNA that it crosslinks to. We did this for a couple of viral proteins shown in pink here on the right side of the slide, as well as a handful of human proteins. For all of them, we could validate the direct interaction. What you see here are not raw read alignments. Those are sites where we see enrichment in our immunoprecipitation relative to a size-matched input control. For the nucleoprotein, as expected, you can see that there's extensive coverage, extensive binding throughout the entire viral RNA genome. For the spike protein, we find a little bit more sparse, but still clear sites of enrichment, and also interesting binding profiles for several of the human proteins that we took an interest in. Before we talk about these proteins in detail, I want to step back a little bit and take a global view on this RNA protein interactome. We started by comparing our data set to previously published data in the same cells. I forgot to mention that we used HEH7 cells after 24 hours of SARS-CoV-2 infection. There is a HEH7 poly-A RNA interactome coming out of Matthias Hens's lab, which basically characterize all RNA binding proteins or all mRNA binding proteins. We find 60% of the proteins that we found were also present in this data. Actually, 66% of the proteins were also present in this poly-A RNA interactome. There was another study at the time that we thought was really interesting, which was a dissection of the proteins associating with dengue and Zika virus RNAs. However, this was done with formaldehyde cross-linking, so this would get you all of these indirect interactors in addition to the direct interactors that we are talking about. However, there's still about 60% overlap. Looking into the functions of the proteins that we found to bind to the viral RNAs, and there are no surprises here. Many Go terms linked to translational regulation or other RNA-centric processes such as nonsense-mediated decay or the co-translational targeting of proteins to membranes are enriched here in addition to viral processes. We then constructed a protein-protein association network based on these direct RNA binders. This is the main network is shown on the left side of the slide here. There are no imputed proteins here. These are all proteins that were detected in our interactome, and they're extensively interconnected, much more than you would expect by chance. And many of these proteins, and those are the ones that are highlighted here, are or have previously been linked to viral infections, which we thought was intriguing and satisfying. About 30 of these proteins are differentially phosphorylated in response to SARS-CoV-2 infection. This is shown on the subnetwork on the top right corner. And below that, about 24 proteins are known targets for approved drugs and compounds. I will talk about both of these things a little bit later in the presentation. First, we wanted to characterize what are the changes in the proteome, in the host cell proteome upon infection. So we quantified or measured protein abundances in infected and uninfected cells. This is a very deep proteome. We quantified 11,000 proteins. By the way, all the mass spectrometry work is done in Steve Carr's lab at the Broad Institute. And we observed a clear upregulation in components of, for instance, the interferon response. We also saw a clear signature of TNF-alpha signaling through NF-kappa B, TGF-beta, and IL-6, so a very broad pro-inflammatory response, which is also consistent with transcriptome studies that people had published earlier. So now we wanted to connect our direct RNA binders to these regulated proteins. So we constructed this protein association network here, where every node is, if it's blue, it's a not regulated protein that binds to the viral RNA. If it's red, it's a regulated protein that binds to the viral RNA. And the connections here are protein-protein interactions, known protein-protein interactions that are also regulated in our proteome measurements. And if we perform a Go-term analysis on these regulated protein communities, we detect that there are many pathways that are emerging as functionally important in the context of SARS-CoV-2 infection. I'm providing some references here if you, in case you want to look this up. For instance, for chromatin organization, myeloid-mediated immunity, Golgi vesicle trafficking, protein phosphorylation, G protein receptor signaling, and protein folding. So we thought that indeed our interactome appears to capture the biology of the virus quite a bit. So with that, we're ready to look at individual candidates that we try to probe for their functional contribution to SARS-CoV-2 infections. We started at the top of our list. CMBP was the protein that was most strongly enriched. We had already confirmed that CMBP directly binds to viral RNA. And we then compared our RNA protein interactome with a, at that time, newly published CRISPR perturbation screen. And what was measured here is basically the guide RNA induced or virus induced cell death as a function of guide RNA expression. And overlaying this CRISPR perturbation screen with our direct RNA protein interactome showed that the protein CMBP that bound strongest to the viral RNA also had the strongest effect on virus-induced cell death. You see in a negative C-score here that indicates that these cells are sensitized to virus-induced cell death. A positive C-score indicates that cells become resistant to virus-induced cell death. So we thought this was really interesting and generated our own CMBP knockout cells, again in these HUH7 cell lines that we had used. And we observed that upon SARS-CoV-2 infection, intracellular viral RNA levels were indeed upregulated in these CMBP knockout cells, indicating that CMBP may indeed be an antiviral regulator. We looked where else the CMBP binds across the transcriptome of an infected human cell. This is again done in HUH7 cells. Most of the protein transcripts targeted by the CMBP protein are protein coding transcript. Binding sites generally are enriched in the coding sequence. And we see this characteristic binding profile with start and stop codons being enriched and signal across the entire coding sequence. And this sort of suggests that this may be, this protein may function in the regulation of translation, which is precisely what had previously been described for CMBP in a PAR-CLIP study in uninfected HEC293 cells by Stefan Juranek's group. If we look at the target transcript level, we see 46% overlap of the targets that we find in infected cells compared to what had previously been reported. A similar binding pattern can be observed here. So we're confident that we're capturing true positive interactions of CMBP here. We already looked at the binding profile across the SARS-CoV-2 RNAs. We're now looking at these sites that are strongly enriched and trying to elucidate any sequence features that could explain why CMBP binds to these sequence regions. With that, we can move on to the next regulator that we dissected functionally, and that is LARP1. LARP1 was strongly enriched in our experiment. We mapped LARP1 binding sites globally, again in infected cells, and we noted this intriguing enrichment of binding sites at the 5' UTR, especially at the very 5' cap proximal nucleotides. In these 5' UTRs, we can detect, this is a de novo motif search here, oligopyrimidine motif sequences, which is perfectly consistent with what had been known and what had previously been shown about LARP1 binding and regulating to host encoded mRNAs, where it's recognizing oligopyrimidine motif sequences similar to the motif we have detected at 5' ends of mRNAs and translationally represses these RNAs, presumably by displacing the EIF4F complex or preventing formation of EIF4F. Inspecting the SARS-CoV-2 RNAs, we see that, again, peaks of very strong LARP1 binding seem to coincide with these oligopyrimidine motif sequences, and especially at the 5' leader sequence, which is shared between genomic and subgenomic RNAs. We see that there is enrichment of LARP1 binding, and there are also sequence motifs that could qualify as oligopyrimidine motifs. However, this is not immediately adjacent to the cap, but we think it's very intriguing and suggestive that LARP1 might regulate translation of these viral RNAs. We then went on and generated, together with Utz Fischer, 4 different LARP1 knockout cells. We showed that in all knockout cells, there's elevated viral RNA expression, as well as elevated production of infectious viral particles quantified by plaque assays. LARP1 overexpression does the reverse. There's a reduction of viral RNA expression, a reduction of infectious virus production. We can rescue this effect in LARP1 knockout cells by overexpressing or complementing these cells with a LARP1 GFP protein and comparing the expression of these LARP1 GFP expressing cells to GFP alone, and we see viral RNA expression is reduced in all 4 of these knockout cells. LARP1 is also phosphorylated, dynamically phosphorylated in response to SARS-CoV-2 infection, and it's a known downstream target of mTORC1. And we could show that, in fact, many of the known phosphorylation sites, this is unpublished data now, are dephosphorylated in response to SARS-CoV-2 infection, and this includes many rapamycin-sensitive LARP1 sites, which would make sense, thinking that dephosphorylated LARP1 can bind to its target's RNAs and translationally repress them. And then finally, for the last couple of minutes, I want to talk about our efforts to use small molecules to inhibit some of these direct SARS-CoV-2 RNA binders. We did this in CALO3 cells, which are similar to HEH7, are naturally susceptible to SARS-CoV-2 infection, but this is a lung cell line. We tried a handful of inhibitors, some targeting direct components of our RNA interactomes, such as ATP1A1, ARP2, as well as DDX3X. We also included some upstream regulators, such as mTORC1. We see that DDX3X only shows a very, very moderate effect at the highest concentration. However, the 3 other inhibitors show a clear dose-dependent reduction in viral RNA levels intracellularly, as well as infectious viral particles. Same is true essentially in HEH7 cells, except for DDX3X, we don't really see an effect. For the others, we see a dose-dependent reduction of viral RNA levels and viral replication and infectious virus. Here are just 2 more RNA binders. PPIA would be a direct RNA binder, TBK1, an upstream regulator of DDX3X, for instance, that is involved in nucleic acid sensing and contributes to interferon signaling. Inhibition of TBK1 only led to a meaningful, consistent effect in A549 cells that have been transduced with ACE2 receptor, not in the other cell lines we tested. So with that, I can move to the summary, and I think we're perfectly on time. We published this study end of last year. This is the first global atlas of direct interactions of the SARS-CoV-2 RNA with host cell proteins. We identified CMBP and LARP1 as antiviral regulators. We mapped their binding sites in the transcriptome as well as on viral RNA, and we demonstrated that pharmacological inhibition of RNA interactome members can diminish SARS-CoV-2 replication, at least in cell culture. There's now more and more screening papers emerging, genetic screening papers emerging, and many of those validate some of the proteins that we found as functionally important. With that, I want to thank my lab. Everybody really got together and contributed to this work. Nora Schmidt here has been the leading author. She's the virologist in my group, and really she deserves most of the credit. I also want to thank Steve Carr and Hasmik at the Broad for working with me for many years now on the proteomics, and also our local collaborators here, especially Jochen Bodem, who got early on an isolate of the virus and enabled much of this work. And thanks to you for your attention, and please let me know if you have any questions.
Thanks, Mathias. That was another fantastic presentation. Like before, we'll take questions in the chat and I'll scan through for hands, but again, it's quite complicated. So if you have a burning question, place it in the chat and I'll read it out. So we have a first one from Ina. Very interesting findings. Do you believe that the altered viral RNA levels when knocking out CMBP or LARP1 are a consequence of the proteins binding to the viral RNA, or a dramatically altered RNA metabolism of the host cell in general? Have you tried to see whether knockdown of these proteins has a similar effect?
So I get the first part of the question. Of course, that's our preferred hypothesis, right? We hope that these are regulators of the viral RNA. But for instance, for LARP1, you could easily imagine that it represses host RNAs, and this is and since it targets top mRNAs, right? So 5' terminal oligopyrimidine RNAs, which are enriched for components of the translation apparatus, this could also lead to a repression of viral RNA production, but not by regulating the viral RNA, but by regulating these host transcripts. So that's definitely something that we need to disentangle. We're currently trying to do this by, for instance, using ribosome profiling to see is there really a decrease in translational efficiency on the viral RNA. Ultimately, we would like to be able to take this down to the level of protein binding and sort of displace these proteins maybe with antisense strategies or similar things. That would be the goal, but will likely be difficult. Right, so I don't get the second part of the question, which was about whether knockdown of these proteins has similar effects on the host cell in general. So I'm not sure what to make with that. Maybe I partially answered it with the first part of the answer.
So yes, thank you. We got a reply. So I think we're still waiting on some other questions. Mathias, in the meantime, I'll ask one. So you had data there showing that there was another antiviral protein, ZAP, that's binding to the RNA. Can you talk a little bit about this? Is the virus in general inhibiting these antiviral factors if they're still binding to the RNA?
I'm not sure if I can say something about a global trend of all of these, but it seems like also from the CRISPR screen that was published that most of these RNA binding proteins actually have an antiviral effect. Some of them are upregulated, some of them go down. For CMBP, for instance, that went up. So we thought the ones that are upregulated are kind of interesting because they may, may be components of specific host response pathways. For SAP specifically, yes, that's an RNA binding protein that has been described to inhibit and actually destabilize or lead to the degradation of various viral RNAs. We're in a collaboration there on sort of trying to tease apart the function of SAP in the context of viral infection. And it does seem to do this destabilizing, and there seems to be a sequence preference that you can find when you map where it associates with these RNAs that it destabilizes. So yeah, that's a very interesting protein as well.
So any other questions for Mathias? Place it in the chat or raise your hand. There's a lot of data there to talk about. Tap, tap, tap. We have another one. So would RAP-MS allow a time-resolved binding profile of RBPs upon infection? Again, from Neva.
Yeah, exactly. That's an excellent question. It's basically what we're doing now. We published a single time point, but which was 24 hours. But of course, it's interesting to look a little bit earlier and a little bit later. Right, so a handful of time points will probably be possible, but much more than that would probably be limiting in terms of the mass spectrometry that we can do. But basically the limiting factor here is really expression of the target RNA, right? So, early on you start to see the RNA accumulate. That's a good time point, but if the RNA isn't there, you won't end up with anything meaningful. Right.
Yeah, and then we have one hand, Neva.
Thanks, Matthias, for this nice talk. I have another question regarding the cell type and infection rates. Basically, also the recent CRISPR screens published back-to-back show very little convergence of the host factors that might have an influence on the viral infectivity, right? So do you plan to also test different cell types? Because we don't know whether HGH7, I mean, the people are also kind of changing the cell models. Have you ever tried comparing it to different cell types and whether direct interactions would be, of at least the major hits would be there or not? Yes,
definitely. So, you know, we moved from HGH7 cell to a cell line of the lung. We used A549 cells that were transduced with a STU receptor, which, you know, is not the perfect physiological model, but this is basically the data set that we have in hands now. However, the more experiments we're doing, it turns out that actually these Calu3 cells are a really good infection model. And I think we might end up trying those as well. And people are doing that. I think there's a study from Alfredo Castello doing similar things in Calu3 cells already. Yeah.
Okay, and then I think we'll take a final, two final questions you got in there just in time. First from Patrick Bonn, could you yet disentangle full-length viral genomic RNA from subgenomic RNA in your pull downs?
Yes, absolutely. So excellent question. And we start doing that and that works for sure. So the best strategy seems to, in our hands, seems to be you can actually use different probes that sort of map to, let's say, the ORF1AB region only and pull out the genomic RNA with these probes. And then after that is depleted, flow in your probes against subgenomic RNAs and isolate that. And that seems to work reasonably well. So, yep, good point.
Okay, and a final question from Akira Onu. Sorry if I missed it. Is there any species-specific difference in binding of RBP to SARS coronavirus 2 RNA? If not, why would the virus retain the susceptible RNA in evolution?
That's very interesting. So we haven't really looked into it, but you know, it's not a human-adapted virus, right? It's the zoonotic virus, so it didn't really have too much time to adapt or change its RNA. But we're seeing these changes happening, right? So it would be interesting to take some of these, you know, more infectious strains and see, are there any differences? Did they respond to some of these, let's call them susceptibilities? So very interesting. We haven't really started looking into it.
Okay, shall we wrap up here? Thank you to the speakers for 2 excellent talks and to all of you for listening, and see you at the next RNA Collaborative seminar.